This lecture is going to cover Chapter 19, discussing the eukaryotic genomes, organization, regulation, and just a bit about evolution. Start first with the discussion of chromatin structure. We've referred to it throughout nearly the entire semester, trying to understand it. And we've always noted that it is based on successive levels of the DNA packing. So how do we start with the linear sequence of nucleotides to make the polymer nucleic acid and end up with it all crunched together? Thank you very much. That was Benny coming to our rescue. He noticed the pen was running low, so he brought a fresh one. Okay. So it's based on successive levels of DNA packing. So with a mitotic duplicated chromosome, we would expect to have the highest level of packing, highest degree of packing, and therefore all the levels involved completely. With the stage of interphase where DNA replication occurs, we would expect it to be unpacked completely, at least in those areas where it's being read for replication by DNA polymerase. So let's use an example. And that's not the only time that we have the condensation or the packing of DNA. In the somatic cell where we have differential regulation, we have different degrees of packing for untranscribed and transcribed genes, the differential gene expression. Well, let's consider the example of the condensed, duplicated chromosome in mitosis. Remember, once the sister chromatids separate, then the sister chromatid that was one half of a duplicated chromosome now becomes a chromosome unduplicated in and of itself, and it's still, initially at least, in that highly condensed form. And if that cell is going to go on to differentiate and not undergo mitosis, then you're going to get the unpacking to the degree where the appropriate genes for that cell are going to be expressed, and it takes on its functional phenotype based upon the expressed set of genes. So this would include these structures. So first, there's the DNA double helix. So if you twist a rubber band, it coils and shortens, right? So just the formation of the helix, we actually shorten the linear length of the DNA polymer. But this produces a diameter, if you will, of 2 nm nanometers. Well, next then we begin to have proteins involved and nucleosomes are formed. We'll talk about those in more detail. And that produces 10 nanometer diameter. So I don't know what degree to which we have shortened the overall length of that DNA polymer, but by bundling it up into little beads, we have created a maximum average diameter of 10 nanometer. Next, there's more coiling. So think of turning that rubber band on a little airplane. As you turn it, it'll coil, and then it'll start forming little clumps, like beads along its length, and then it just starts really supercoiling. And that's what we're beginning to see here. So we get 3 times the overall average diameter, producing fibers with 30 nanometer fiber. And then it bunches up, and we get 300 nanometer fibers. These are both plural. And that gives the looped domains. So as we get our coiling, there are points where little bits of the DNA form these loops shooting off from the main backbone of the structure. And it's the average overall. I drew these loops all the same length. They're not all the same length extending from the backbone. The average would be 300 nanometers. So it's at the 300 nanometer fiber stage that the loop domains appear. And then it'll begin to fold, where it fold up like this, kind of like an accordion, still with the loops coming off it. So it's just scrunching together as like an accordion. And then you get a new, a little more than doubling of the overall average width to the 700 nanometer fiber stage. So it folds like an accordion, and this is when it is maximally compacted. So all these levels of packing are involved in producing the most condensed chromosome. So over here on the left-hand side, we'll put an arrow, and of course make the note that it's, as we progress down that direction, increasing. We see increasingly higher levels, H-I-G-H-E-R, levels of organization. So the chromatin structure is much more complex as we proceed through, obviously. Well, let's just take a moment and consider the nucleosomes. So the nucleosomes are the beads. If we looked at the 30 nanometer fiber, at the nucleosome stage, there'd be strand of DNA, and then a nucleosome, strand of DNA, and a nucleosome. These would be the first little kinks that would show up in our rubber band as we're winding it. So these are called like beads on a necklace, the nucleosome beads. So in the nucleosome, each of those little beads, this is the first level of protein interaction with the DNA. So the DNA is wrapped around histones. A special type of nuclear proteins. So as I said, that's the first level of the DNA protein interactions. So you can see I'm abbreviating protein as PROT. And it consists of, to quote the text, of two molecules and each is of four types of histones. So obviously the illustrations in the textbook and the PowerPoint slide presentation shows that much better than I could ever draw it, so I'm not going to attempt to do that. So there's two sets of four histones. So this means that there are 8 histones, histone proteins, and we'll talk about these proteins a little bit more, per nucleosome. So there are two molecules, basically 4 of the histones clumped together, and then you have 4 different histones that are chosen from, basically. But overall, it end up with 8 histone proteins per nucleosome. Well, let's talk about the histones now. So as I said, each nucleosome consists of 2 molecules, each of 4 types of histones. So the 4 types of histones in the nucleosomes are H2A, H2B. So these are, H is obviously for histone protein type 2A, type 2B, H3, and H4. So those are in the nucleosomes. And of course, there is a histone 1, histone H1 is not present within or inside or in the nucleosome bead. It instead is involved with or involved in the formation of a higher level of chromatin structure. So it binds to the DNA just adjacent to the nucleosome. So it begins to contribute to the transformation, if you will, or the change in the level of chromatin structure from that of the nucleosome to that of the 30 nanometer. So it's one of the additional proteins that contributes to that transition. It's also important to note, now that we've mentioned H1, 2A, 2B, 3, and 4, that the total mass of histone in chromatin is approximately, so this little wavy line, I use that as a notation for approximately equal to, so over an equal sign, it means approximately equal to the mass of the DNA. Now, in that three-dimensional nucleosome, which I can't draw, I'm not going to try to, you've got the DNA wrapped around 8 of these histone proteins forming 2 molecules, each of 4. And the amino ends, remember the histones are proteins, so they have an amino terminus, and the stretch of the amino acids toward the amino terminus is called the amino end. So the amino end of each histone protein, each of the 8 separate ones, so there's 8 little amino end tails sticking out in a nucleosome. So the amino end of each histone protein extends outward from the nucleosome. So you might be able to picture having eight big oil drums all wrapped together. So if this was one oil drum, and we had 8 of them, and then you wrapped a coil around all 8 of them like that, and each oil drum would have a spigot sticking out. So there'd be 8 of those in there. I'm not going to try to fill them in because it would look horrible, but this is what we're talking about. So these are portions of the histone proteins available for modification, and we'll see that becomes very important. So that little thing sticking out is called the histone tail. PAIL. And as noted, can be modified. One example is when it's acetylated, an acetyl group added to it. So when that tail is acetylated, this promotes the loose chromatin structure. So in other words, this leads to the availability of that portion of the DNA for transcription. So we said that in a daughter cell, as an outcome of mitosis, it's going to go on to differentiate into its functional state, functional phenotype. One of the first things we'd have to do is appropriately acetylate the histone proteins to begin to loosen the chromatin. I'm sure there are other things that occur before that maybe, but ultimately we'd have to do that to loosen it up enough to expose the DNA so that the genes could be expressed. All these histone proteins are found in mammals. And in other animals, and plants. So in other words, eukaryotes. So this is just kind of summarizing it, but we know that it's globally throughout all of the eukaryotes. Well, next then, let's talk a bit about the regulation of chromatin structure. So as we noted, the acetylation of the histone tails is included here. So in the unacetylated or non-acetylated form, we get the progression from nucleosome stage to the 30 nanometer stage. With acetylation, the addition of an acetyl group to this, the histone tails, we begin to get the loosening. Well, there's another type of functional group that can be added to the histone tails, a methyl group. So methylation of histone tails is another example, another mechanism that we have more understanding of now. And this leads to condensation of chromatin. So it may be, I don't know if the same amino acids or parts of amino acids are targeted by the acetylation as they are for the methylation, but we could kind of picture that you'd have to add methyl groups when the acetyl groups are not there to condense the chromatin. So I don't know if they interfere with each other, but it seems that at some level, indirectly at least, they would interfere. If you had a lot of acetylated histones and the loose protein chromatin structure, then it's possible that just adding methyl groups wouldn't coil it up tighter. You'd have to deacetylate first and then add the methyl group. Anyway, they are reversible is the important thing. While histone tails are not the only thing that can be methylated. Actually, some of the nucleotides in the DNA can actually be methylated. So there's DNA methylation as well. So not only regulation at the level of the proteins binding to the DNA, but modifications by putting functional groups directly onto the DNA itself. So this occurs with certain bases in the DNA, and of course it's after synthesis. So during replication, you synthesize the new strand. After it's synthesized, then you begin to modulate it, modify it, and the addition of the methyl groups is one of the first things that can happen. And this will occur usually with the cytosine nucleotide. The DNA methylation is especially common excuse me, with inactive DNA, meaning not being transcribed. Inactive DNA. So therefore, we would expect the most methylation to be present in the DNA within an inactivated mammalian X chromosome. None of the genes on the inactivated excuse me, mammalian X chromosome are expressed. So that's an example, the complete inactivation with high degrees of methylation, as well as condensation. So many factors, many mechanisms are involved in completely inactivating the X chromosome. Well, it is possible to have activity at methylated DNA. So if there is activity of methylated DNA, then the gene expression would have, or those genes would have turned off, or as an outcome, we would have turned off or just slowed down the process of transcription. Right, so part of our focus in this chapter is on differential gene expression and what are the mechanisms that can regulate that. So even though we're talking about chromatin structure, we're already relating it to the differential regulation of gene expression. So now let's begin to think of the ways that we can regulate gene expression. So everything we've talked about so far, little arrow shooting up, of course, these are the highest levels of differential gene expression. In other words, is the gene in its promoter region available even to have the transcription factors and RNA polymerase to bind to it? It's all coiled up into a nucleosome and then even tighter after histone 1 is there, and you've got no acetyl groups and you've got methyl groups on the DNA and on the histones, then the transcription factors and the RNA polymerase can't even get to the promoter to bind to it to initiate transcription. So that's the highest level, at the level of protein interaction with the DNA. It's also important to note at this point that approximately, there's our wavy line again, approximately 2%, that is, the proportion of DNA in the human genome codes for protein. Not all genes code for protein. Some code for functional RNA. Ribosomal RNA, the miRNA, siRNA, and a few others that we've been discussing. So the remainder, approximately 98%, the remainder is mostly non-coding sequences. Remember, we're talking about eukaryotic DNA here. This would not be the case for prokaryotic DNA. There isn't much extra DNA present in those genomes whatsoever. However, in both prokaryotes and eukaryotes, gene expression is largely regulated by transcription. So that is in prokaryotes and eukaryotes. So when in prokaryotes and in eukaryotes, the DNA's the promoters available, transcription factors can bind to it, then we can begin regulating gene expression. In the prokaryotes, it's going to be there. It's not all coiled up. In the eukaryotes, we have this various levels of excuse me, protein interaction with the DNA, or methylation, functional groups added or taken away from the DNA itself. But once you get that condition established for a particular cell, what genes are going to be expressed, then it's control at the level of gene expression. While we're right here, and to make the transition over to illustrating a eukaryotic gene, remember that prokaryotic genes do not have introns, do not contain introns. Actually, it's just one gene after another, and the DNA sequence within a gene was available to produce messenger RNA directly without any RNA processing. And then that way you could have translation of the message occurring at the same time it's still being produced off the DNA through transcription. Well, in eukaryotes, that's different. You've got to process the RNA. So let's just illustrate a eukaryotic gene to start defining some of the pieces and parts of it. So I'm going to put a line all the way across the piece of paper. Put a vertical line here, and that's the start site. That's where transcription would begin. S-T-A-R-T. That's the start site. And the eukaryotic genes have exons. I'm just going to put 3 of them. So it would be exon 1, intron 1, exon 2, intron 2, exon 3. Somewhere here would be the information for the poly A tail. And then somewhere farther on, so these double lines here mean that there's a big gap. There's going to be the termination region. Termination, I'd really fit it in that region. All right, so initiation, elongation, and then ultimately termination of transcription. Well, just so this is downstream of the start site. And obviously the other direction is upstream. So we're using the start site as our reference point. And we could say upstream of exon 3 is intron 2, but we're saying upstream of the start site is the promoter region and areas of control elements. And then downstream we find the exons and all the things that we've labeled here. So the immediately initial portion of the upstream DNA is called the promoter. The TATA box is there that helps trigger the binding of the RNA polymerase when everything else involved helps it. We're going to put a split up here, so there's a lot of DNA nucleotides in between these two tilted lines, forward slashes. And just immediately upstream of the promoter is called the proximal control elements. They're close to it. They approximate it. So I'll zoom in on this part now. So there's the promoter and then the proximal control elements. Some publications and texts will refer to these as regulatory elements or response elements. The textbook, the authors decided to go with control elements, and they can either be a regulated or a response element, but that's not a problem for us. Then as we go farther upstream, there are these enhancer sites, enhancer region, and there's just certain portions of the DNA that are really the enhancers up there, specific for binding particular proteins. But altogether, this is called the distals, farther distance to this point upstream. These are the distal control elements. So we'll talk about activators and repressors that can bind up here. But hormone receptors, ligated hormone receptors, might bind here in the proximal control elements. Proteins activated by cyclic AMP. We've talked about these intracellular signaling mechanisms in Chapter 11, I believe it was. So we also talked about changing gene expression due to signaling between cells by chemical messengers. And it's by actions mostly on the proximal control elements that those occur. So just to zoom in the start site, the exons, introns, well, I put poly tail, I forgot to put the A in there, poly A tail, and then somewhere far downstream, the termination region. That's a poly A tail. So this area tells the nuclear systems to add the poly A tail there. So obviously, we get the first event occurring transcription. Produces what's called the primary transcript. First one, it includes, it's the RNA molecule, but it includes the same, the complement of the sequence all throughout the coding region of this gene. So from the start site on, that's the true encoding region. And then after that, there's, of course, steps to produce the messenger RNA. collectively these are called RNA processing. And they've been discussed in another chapter about protein synthesis. So we'll build on these different levels of regulating gene expression. We're beginning to see we can obviously control the DNA packing, transcription. Regulation at transcription is the most prominent means for differential gene expression once you get to that point. But RNA processing can be regulated, and we'll talk about some other levels, all the way to the point of the protein living its life and being removed or degraded, dying, in other words. So let's consider a couple definitions here real quick. So we have two that are DNA. We saw the enhancers and the distal control elements way upstream, the promoter. So these two are actual DNA sequences. And I'm not going to define all of these. Promoter we've talked about in the prior chapter. An example of enhancers is that the function is transcription control of gene expression. When we talk about the proteins that can bind to it, we'll see how that works. So there can be two types of proteins that can bind to the enhancers: an activator. I'm going to leave space below it to define it, and repressor. So these are proteins, and by definition, any protein that binds to DNA and regulates gene expression or transcription, it's called a transcription factor, or TF, transcription factor. So these are examples of some transcription factors. The hormone receptors are examples of transcription factors. Like the steroid hormone receptors. They're not membrane bound, they're inside the cytoplasm or the nucleus. And when ligated, then they bind to DNA and change gene expression. So they're a different family of transcription factors. So the activator and the repressor, as I said, are targeting these distal control elements at the enhancer. So an activator is a protein that binds to enhancer and, of course, stimulates transcription. So it's pretty obvious from the name of it. Well then, repressor is also a protein. It's a protein that can inhibit transcription by blocking the binding of positively acting transcription factors put TFs for transcription factors. So the repressor can bind to the DNA and can inhibit the binding of other proteins. So it blocks the binding of those, especially those that are positively acting. In other words, they would increase or enhance or stimulate gene expression at the level of transcription. That may not be the only type of repressor, but that's a good example of how they work. So then let's summarize the levels of regulation just quickly. And this is regulation of gene expression overall, from signaling to the gene to the death of the protein product of that gene. So we'll have an extensive list here. So we said it can start with the degree of DNA packing. And the DNA packing can actually be affected by transcription factors too. So we can just loosen the coil of the DNA a little bit to expose more upstream portions of a gene next to a nucleosome so that we can have more complex regulation through other transcription factors. And one way that we can loosen the DNA packing a little bit more near a nucleosome at the edge of a gene is intracellular signaling that results from the extracellular signals from other cells, like growth factors. So some of the transcription factors that are used by signals from other cells can actually demethylate DNA or act on maybe even the histone proteins. Okay, so subsequent to that, we know we're at the level of the regulation of transcription, as we just discussed. Then there's the RNA processing that we listed before. Then, of course, as far as translation would be concerned, we're looking just at the messenger RNA, but there's other RNAs that are involved, but we want to think of it in the most direct form here. Then we get the messenger RNA in the cytoplasm. Right. And with the messenger RNA, we can have translation occur. So there's factors that impact how impact if transcription will be initiated on a messenger RNA, and there are mechanisms that regulate how many times the messenger RNA will be used by ribosomes to be translated. So in other words, you don't just have a message that becomes available, one ribosome goes down it, makes a protein, and then it's done. It's a whole range of maybe just being used once to being used a zillion times. I don't know what the limits are. I don't know if anybody does know. But what happens is, in that process, you've got this functional molecule having a life cycle, so it'll be produced, as we saw, and then it performs its job, and then at the time it's performing its job, the pool of messenger RNAs undergo a rate of degradation. And we'll come back and make another statement on this in just a moment. Then, of course, at translation, you get the protein made. As the protein's being made, it's being modified, so there's post-translational processing. We talked about the primary, secondary, tertiary, and quaternary structure of proteins, and, of course, activation by being phosphorylated or inactivation by being dephosphorylated. So those are all parts of post-translational processing. The functional expression of a gene is what we're thinking about now. You don't just get the protein, but you get some function out of that protein, right? So the post-translational processing, other than primary, secondary, tertiary, quaternary structure, which is part of getting the protein geared up to function, there might be some other means for causing it to become functional. Let's just call it activation. So protein activation. So it's finally functional. And then, of course, an ongoing process against the entire pool of activated molecules. You get inactivation. Or degradation, right? Well, it includes degradation. So we had used an example in an earlier chapter of the cell signaling mechanisms where you can activate kinases by phosphorylating them, and then you inactivate them by dephosphorylating them. And just because a kinase is dephosphorylated doesn't mean that it's immediately going to be degraded. It has to be worn down, and we also talked about the destruction methods of destruction of worn out proteins. Ubiquitin binds to them, and then it goes through a process of destruction or degradation within the cell. So inactivation is not only degradation. There's other ways to inactivate molecules. Just like the feedback, if you've got 3 enzymes, the final product can inhibit the first enzyme by inactivating it. And that's one form of inactivation also. But it doesn't mean that protein is going to be degraded necessarily. Once it's worn out, then it will be degraded. So let's make that statement about RNA degradation. So in a cell, should be more of a space there, the amount of protein made using a given messenger RNA molecule So any one given messenger RNA, so imagine you are a messenger RNA molecule. We're considering you at this time. It depends, at least partly, on the rate at which messenger RNA is degraded. So if you're a messenger RNA molecule produced in a cell, and for some reason the mechanisms for degrading messenger RNA is elevated in that cell, then the amount of protein made from you by ribosomes is going to be reduced in a cell where you could hang around longer and interact with more ribosomes. So the point we're making is that every step along the way, there are known and unknown mechanisms for regulation. So you can look in the scientific literature at many studies where they've measured the level of messenger RNA in a population of cells, and they say gene transcription is high if the message level is high, and translation is high if message is high also. We now know enough to challenge that and say that the only conclusion you can draw if you perform a study of, say, 2 cells, one from a normal cell and one from a cancer cell, if the cancer cells have higher levels of messenger RNA, that's all that it means, that the messenger RNA level is higher in that type of cell than in the normal cell. And until you examine all the other features of the mechanism, you can't really expand beyond that. What about the protein levels? Are the protein levels increased? What about the levels of active protein activity? Are those also increased? Then we have a better feel for the functional differences between these 2 different types of cells. So let me get off my soapbox here. So let's generalize a bit. So after the point where we have the initial transcript, the transcription event, that transcription event that gives us our first, and remember in eukaryotes there's the RNA processing. So everything that can occur on our list after that transcription is grouped together collectively called post-transcriptional regulation. I'm going to be able to fit it. Post-transcriptional regulation. So even the post-translational modifications are included under this because it happens after transcription, that initial transcription event occurred. So let's consider some examples. Alternative splicing we've talked about. Right, and that occurs in RNA processing because we have the introns and the exons. You have to splice, use the splicing procedure to get the introns removed and the exons fused back together, or their representation within the messenger RNA. We talked about RNA interference. Right, I showed you some articles on that in the beginning of chapter 18 there. We consider the messenger RNA life cycle. Right, and we talked about the translation of that, but let's make the more direct statement of initiation of translation. So we had said that if you were a messenger RNA molecule, the amount of protein produced from you would depend in part on the rate of messenger RNA degradation, but also the rate of initiation of translation would be the other part of it, would be one other part of it, right? And how many ribosomes, therefore, could get onto that messenger RNA and translate it. And then also we'll just put it together as the life cycle of proteins. Right? So translation, post-translational processing, activation, inactivation, including degradation, ultimately broken back down to the raw material, the amino acids. Well, now let's consider what happens when cells become cancerous. So cancer results from so it's all at the level of regulating gene transcription, or we now understand that a big part of what happens in cancer cells is that you've got changes in the way that gene expression is regulated, especially those that affect the cell cycle control. So the biggest problem with cancers is that they can spread. They just keep growing and growing and growing. They are invasive, and they can become migratory. They can break off from the primary site and distribute through our body fluid systems, lymph or blood, and set up secondary sites of tumors. The metastasis, of course. Let me go to a new piece of paper here. So what are some of the mechanisms then whereby some time bomb genes called proto-oncogenes are converted to oncogenes. If you hopefully don't have any direct experience of going to an oncologist or an oncology center, But certainly, I'm expecting that you have heard of oncology centers or an oncologist, and the onco, of course, means cancer. So these are these proto-oncogenes are genes that can be converted into oncogenes and promote the transformation of a normal cell, normal eukaryotic cell, into a cancerous cell. So the first mechanism, there are three of them, is the movement of DNA within a genome. The gene goes from one place to another, so it can be through chromosome translocation, chunks of chromosomes, or gene transposition. So you can have translocation of parts of chromosomes, or genes being changing their position within the genome. Then the proto-oncogene is in a different environment as far as regulation of gene transcription is concerned. So we're talking about converting an inactive oncogene to one that becomes active and somehow then supports the transformation of a normal cell to a cancer cell. And as far as I know, it's only the cells with mitotic capabilities that have this type of an effect. They already are capable of undergoing the cell cycle, the cell division cycle. So that's the greatest concern. They're dividing anyway, so if something happens so that their mechanisms for regulating their cell division become uncontrolled, get out of whack, then they just keep dividing and dividing and dividing. So a post-mitotic functional cell, like a cell in the lining of our intestine or on the surface of our skin, is less likely to become cancerous because it's not going to divide anyway. So you'd have to change the way it is differentiated to the point where it could undergo division again. And that's unlikely. The cell's more likely to live its life. Like, how often are we shedding surface cells from our skin? All the time, constantly. The lining of our gastrointestinal system from the stomach to our anus is completely changed within 4 to 6 days. So the functional cells have a short lifespan. So it's really the cells that are capable of mitosis, the reserve cells that can undergo these types of changes that we're talking about. Our reserve cells live their entire life with us. If we lose our reserve cells, then the functional cells we have that are used to being replaced in short periods of time won't be there, and we won't live very long. Obviously, our neurons, our skeletal muscle cells, and our cardiac muscle cells, for example, are post-mitotic cells that have got to live as long as we do. If they die, we can't readily replace them. What happens if somebody has a blockage of an artery in their heart? Part of their heart muscle cells die and they can't be replaced. It becomes fibrotic, a scar forms, or it can even become calcified, but you don't grow new heart cells. So that's a big difference between the two. The reserve cells, where our cells are replaced, are critical, and they have to live as long as we do. So you can end up with, the amplification here means generating multiple copies of a gene. So the ways that this can occur are discussed in the textbook and the PowerPoint presentation. There's a few different ways. And then third is point mutation. Point mutation can be either in the control element or an A control element. We talked about those already. on the promoter or on the proximal control element or the distal control element, something that alters the rate of the expression of that gene, or it can be in the proto-oncogene itself. Generating a monster protein that either is very active or somehow resists degradation, and it lives longer and has a longer half-life, right? It lives longer and stimulates changes in the cell for a longer period of time. So let's kind of summarize that. These collectively lead to excess growth stimulating protein, but let me put in here just signal, right? You may have more protein just from a higher degree of transcription and translation for this gene now. So it may be just an increased amount of the protein. So there's more to become active, and it's around longer, overwhelms the rate of degradation. So there's more signal present. Or you might have, as with the proto-oncogene point mutation itself, you might have generate through a mutation a hyperactive molecule. So it binds the receptor and just stays there and keeps the receptor stimulated. Or maybe it is degradation resistant. Doesn't matter, the total, signal. We think of the signal telling the cell to do something. There's more signal. Well, in addition, there are possibilities, as we see, with interference with the normal cell signaling pathways. There's one possible outcome of what we just discussed. One example is a tumor suppressor gene, P53. So that's the DNA. Right? So it's a tumor suppressor gene. In other words, the brake is on all the time. If you are driving down the road and you got your foot on the brake, it's going to keep you slowed down, right? Take your foot off the brake, you're going to go faster. Don't recommend it. Your brakes will be burned out real fast. So this gene drives the synthesis of a protein, obviously, and the function of this gene with its protein is pretty complex. It slows down the cell cycle, right? So that can be occurring only in cells that are undergoing mitosis in the first place. So we're back to the idea of the reserve cells. In a post-mitotic cell that's performing its life function, it's not undergoing the cell cycle anyway. It's totally repressed and shut down, so it wouldn't matter if the P53 gene was changed in that one. But these are normal functions. So it is an activator of other genes. So it can work as a transcription factor. And by slowing down the cell cycle, or even by inhibiting the cell cycle, actually, this is even more severe. So it inhibits the cell cycle and prevents the cell cell from passing on mutations due to DNA damage. So in this case, it gets even more serious. It doesn't just keep a slower cell cycle. It will actually inhibit the cell cycle. Tell that cell that has DNA mutation, don't divide, don't spread. We don't need more of you. And can even proceed to the point where it causes cell death. So yeah, it's a tumor suppressor gene, everything we talked about. Well, another example is the RAS proto-oncogene. So be sure you pay attention to when the gene is being discussed directly and the protein is being discussed indirectly, or they're kind of considered collectively. This gene, along with its protein that it is capable of having produced, function this way or that way. So the RAS proto-oncogene produces or encodes for the RAS protein. And normally the RAS protein is involved in, need to back up here, get more space. It's part of intracellular signaling mechanisms, so it's involved in relaying a signal from a growth factor receptor. Okay, so growth factors bind to their receptors on reserve cells and regulate the rate of mitosis. And so if you mess up at this point, you can get cancer-causing forms of the protein. And they are usually hyperactive, as you might expect. So if this RAS protein is involved in the intracellular signaling due to an extracellular signal, a growth hormone, that's telling the cell to grow and reproduce, if this intracellular signaling protein becomes hyperactive, it just continues to tell the cell to divide. So I don't care if there's a growth factor out there or not. I'm telling you to divide. I'm wrong, but I'm telling you to divide. That's my mutation. That's where I've changed from a proto-oncogene to an oncogene. I now promote cancer-producing cells, or cancer type of cells. So then, proto-oncogenes become oncogenes through those mechanisms that we talked about. And of course, the outcome is some type of a cancer. Now, what we're considering here are tumors, metastatic tumors, not the benign tumors necessarily, but some of these might be involved in producing benign tumors, but it's rare that individuals will die due to benign tumors, so our real concern are the malignant tumors capable of metastases. Any of you ever seen the television show, they removed, I forget, was it 300-pound benign tumor from a woman's abdomen or something like that? Still didn't kill her, it just is amazing. Absolutely amazing. So we consider this as being the presence of potential time bombs. You might recognize this as one of the quiz questions for this chapter. eukaryotic cells. And we explain this by saying that proto-oncogenes normally help regulate cell division. So if you regulated the production of cytoskeleton in a terminally differentiated cell, wouldn't really matter as much as if you were a gene that is involved in regulating cell division within a reserve cell. So that's the point that's being made here. So they're a clicking time bomb. Whatever might cause the changes, we listed the three of them, that would convert a proto-oncogene inside our reserve cells, any of our reserve cells, to become an oncogene, then we are very susceptible to cancer, right? So proto-oncogenes can code for proteins associated with cell growth, as we said. So again, this is consistent with my emphasis, gene to protein. So the gene doesn't do it, it's the protein that does it. Proto-oncogenes can be involved in producing proteins for cell adhesion. So if there's a change such that the cancerous cell doesn't stick to the surrounding connective tissue or extracellular matrix like it's supposed to, now it just balls up, lets go, and floats off into your lymphatic tissue or your blood, and then sets up residence somewhere else. Or it might become motile, to where it can decide what parts of the extracellular matrix it wants to stick to reach out and pull itself along like an amoeba moves. So this is relevant in two ways. If they became stickier and stayed right there, they'd never become metastatic, and you could find the primary, the site of the tumor, take it out cleanly, and then, well, it'd be benign then, wouldn't it? So that's why that one is important. Not all of them, but some of them can be involved in that. So similar to, they are similar to oncogenes found in retroviruses. So it makes us wonder where they came from in the first place, but we see that not all of them came from there. And they can code for proteins involved in cell division. I got just a couple more pages. Oh no. Hey. Yeah, I do. Just a couple more pages, so let's push on. So then we can talk about, from the individual perspective, whether it's you or your pet. I don't think they're testing for it in pets, but probably could find somebody that would be happy to take your money to do it. So for predispositions, it's possible to have a genetic test, and some of the prime examples that we know of are the APC gene for colorectal cancer, and second is the BRAC1 and the BRAC2 genes for breast cancer. So some cells are tumor cells are very responsive to gonadal steroid hormone stimulation, such as prostatic tumors, cancers, obviously in males, and breast cancer in females. So in the breast cancers that have these proto-oncogenes involved, they tend to be very sensitive to estrogen. And the treatments are geared along those lines, following surgery if surgery is indicated. So eukaryotic genomes then have the many non-coding DNA sequences. In addition to the genes, we mentioned the 2% before being the genes that encode for proteins or functional RNAs. And thus, the most prominent component of DNA are actually transposable elements. Be an S, transposable elements, of course, and their related sequences. There's a little bit else that goes along with them. Well, if the majority of our DNA in our genome is made up of transposable elements, and let's consider transposons. There's parts of the DNA that can be moved from one location to another. The varieties are discussed in the text and the PowerPoint presentation. But just in general, what they might do if they move, transpose to some place, they may prevent normal functioning of a gene. We talked about converting proto-oncogenes to oncogenes. It may increase production of a particular protein. Or it may decrease production of a particular protein. So the sequence of our DNA for expression of a gene is such that we can regulate the expression of that gene and the amount of protein produced by it. Interfere with the normal functioning of that gene, you can get more protein produced from that gene ultimately, or less. But what it will not do, what transposons do not do, is they do not change, they do not reduce the amount of DNA within cells. It may add to it a little bit, as I understand it, but it's not going to reduce the amount of the DNA within a cell. Well, then let's talk about genes a little bit more, particularly the multi-gene family. So we had stated before that not all the genes for biosynthetic pathway may be on the same chromosome. They're not in tandem as they are in the prokaryote genome. But there are some examples of multi-gene families where the genes are close together or even in tandem on a length of DNA within a particular chromosome. So the multi-gene families, multi-gene families are a class of DNA that codes for the three largest ribosomal RNA molecules, as one example. And this is the example where our genes can look like those of a prokaryote. They're virtually in tandem. So let's consider this in a little more detail. For these multi-gene families, when they're made up of identical genes, so repeated copies of the same gene all together, that's when it will always code for RNA products, such as we just talked about for the ribosomal RNA. Okay, so it's repeats of the same gene to produce, just to pump out the RNA when the RNA is the product. So you don't have to have an extremely elevated transcription rate to get a lot of that RNA. You just have multiple copies of the same gene. So it's the identical gene. But when non-identical, genes, then these will be families of proteins. And of course, the primary example used was the hemoglobins. And I refer you to the table in the textbook where it shows that the alpha major family has two subs. I still don't remember all of my Greek letters. I remember alpha and beta here. And then under the beta, there are three of them listed. So it's an extensive lineage of families of globin proteins that have evolved as the gas-carrying proteins, particularly, obviously, for carrying oxygen, as we know. Such that we have, and they are heterotetramers, so there are four of these subunits together to form the functional protein, with the heme group in there. So there's four globins and a heme group, so that it sets up the structure to have a high affinity for oxygen under the appropriate environmental conditions. All the environmental conditions for the embryo and for the fetus, once it gets past 8 weeks of development, there will be different forms of the heterologous tetramer in the embryo and the fetus forms, and they will have a higher affinity for oxygen, just put O2, than in adults. So what we can say here then is that in this instance, we can see non-identical genes can produce different versions, in other words, isoforms, right, of proteins, isoforms of globins during development. So different stages in our development. We need higher or lower affinities for oxygen. So we express different genes throughout. So once we're out and we're air breathing, the amount of oxygen that is available to the hemoglobin in the lungs to bind to the hemoglobin is much greater than what has to be absorbed across the placenta. So you have a higher affinity for oxygen in the fetal, embryonic and fetal forms than in the adults. Now I've just got one final thing, and thank you for sticking with me on this. Under the evolution, I just wanted to make a point about exon shuffling. Seemed kind of interesting. So we can make a list of 6 genes and consider some exons by letter. So 1 is A, B, C, D. 2 is A prime, B prime, C, and D. And the prime means that it's been changed in some way, so it might have been replaced by a similar gene that's essentially the same or virtually the same, but differs somehow. So not all these are exon shuffling. These are just some examples of how we might illustrate exons that have been changed through evolutionary periods. And 5 is A, B, C, D prime. And then 6 is E, F, B, and G. So, that's supposed to be there. So from this list, I would say, obviously, number 4 happened from exon duplication. Somehow we ended up with an extra exon within this gene. It could be through, it's obviously through recombination events. Gene number 6 is obviously mostly unrelated to the other genes. Obviously, except for the presence of exon B. And therefore, it's most likely a product of exon shuffling. So somehow it was translocated from a gene ABCD with those exons into one that was EFG, and now it has an extra exon in it. So this is an example of exon shuffling. It gets shuffled from one gene to another gene where it's not supposed to be. But you can have the replacement of exons by virtually the same genes through other mechanisms, and still can change the protein a little bit, but it's not as drastic a change as what you'd get from exon shuffling. So that completes our discussion of Chapter 19. I really appreciate your time and attention.